Hi dear students, welcome back to Sharon Zoology. See, in this session, I'm going to explain some previous questions from the chapter Molecular Basis of Inheritance. Okay, the first one. The final part, the final proof for the DNA as a genetic material came from the final proof of DNA as genetic material came from the experiments of. Let's see. See, we know the experiment for DNA as genetic material was conducted by Frederick Griffith in 1928. Then Griffith experiment was experimentally proved by Avery McLeod McCarthy in 1944, and the final experiment was conducted by Hershey and Chase. So let's see the statement from our NCERT book. Okay, look at this statement. See here. The genetic material for DNA. The unequivocal proof that means the final proof, the unambiguous proof, the unequivocal proof of DNA, that is genetic material, came from the experiments of Alfred Hershey and Martha Chase in 1952. So this is the final proof of DNA as genetic material. So the final proof of DNA as the genetic material came from the experiments of Hershey and Chase in 1952. Hershey and Chase in 1952. So here the answer is option 1. If there are 999 bases in a RNA that go for a protein with 333 amino acids, and the base at the position 901 is deleted such that the length of RNA become 998 bases. How many codons will be altered? Let's see. See here, 999 bases in RNA code for protein with 333 amino acids. If the base at 901 position is deleted, see here, if the base at 901 is deleted, then the first 900 bases will be normal. Total of 999 bases are there. Okay. If the bases at 901 position is deleted, then the first 900 will be normal. They will not be altered. Whereas the bases after 901, the bases after 901 will be altered. That means about 99 bases will be altered. The 99 base, the rest of the 901, the remaining 99 bases will be altered. Okay. Yes. 3 bases stand for 1 codon. So 99 bases means 33 codon will be altered. Okay. So 3 bases, we know 3 bases stand for 1 codon. So 99 bases means 33 codon will be altered due to the deletion of 1 bases at 901 position. As a result, the answer will be option B, 33. Here the answer will be option B, 33 is answer. Okay. Next question. During DNA replication, Okazaki fragments are used to elongate. Okazaki fragments. See here the options are the lagging strand towards the replication fork, the leading strand away from the replication fork, the lagging strand away from replication fork, the leading strand towards the replication fork. See, we know in DNA replication, both of these strands act as template. So template, the parental strands are template. Both of the strands act as template. But in one strand, that means the strand lies in 3' 5' direction. The DNA synthesis is continuous. So this is the continuous strand or the leading strand. Whereas in opposite strand, that means the strand lies in 5' 3' direction. 5' 3' direction will be discontinuous strand. And this strand is called as lagging strand. See, in this lagging strand, These short fragments formed in this discontinuous strand or lagging strands are called as Okazaki fragments. These short fragments are called as Okazaki fragments, and these Okazaki fragments are move away from the replication fork. Here this is the replication fork. These Okazaki fragments move away from the replication fork. So here the answer is the lagging strand towards replication fork is wrong. The leading strand away from replication fork is wrong. Here the answer option C, the lagging strand away from replication fork. The lagging strand away from replication fork. So answer will be option C. See, the process in which DNA is replicated and two copies are synthesized is DNA replication, we know. It involves the formation of a replication fork. The Y-shaped replication fork is formed. The two strands of the DNA helix separate and the new strands are formed on the original strands called as template strand. The strand which is synthesized in 3' 5' direction is leading, whereas in opposite direction is the lagging strand. The lagging strand contains Okazaki fragments. This strand is synthesized away from the replication fork. Thus the correct answer is option C. Next one. Which of the following RNA should be the most abundant in animal cell? The most abundant in animal cell. Here, the ribosomal RNA is also called as rRNA that provide the site for protein synthesis, and it forms 80% of the total RNA content in a cell. rRNA, the ribosomal RNA forms about 80% of the total RNA content of the cell. Hence, this ribosomal RNA is thus the most abundant RNA. So here, which one? Option D is the answer. The most abundant RNA in animal cell. Answer is option D. Next is spliceosomes are not found in cells. So let's see, the options are fungi, second option animals, third option bacteria, fourth option plants. See, we know spliceosomes are found in eukaryotic cell. They are found only in eukaryotic cell, not in prokaryotes like bacteria. So here the answer will be option C. Let's look here. Spliceosomes are used to removal of introns during the post-transcriptional processing. That means after the formation of heterogeneous RNA, this hnRNA, the processing will be takes place. And during that processing, the introns are removed by this spliceosome, removal of introns and joining of exons. That process is called as splicing by the spliceosome. And this mechanism is seen only in eukaryotic cell, not in prokaryotes. So it is. So answer is option C. Bacteria lacks spliceosome. Okay. Next. Association of histones with the nucleosome indicate. The association of the histone H1 with the nucleosome indicate. Options: DNA replication occurring, the DNA is condensing to chromatin fiber, the DNA double helix is exposed, transcription is occurring. See, the answer is option B. The DNA is condensed into chromatin fiber. See, the DNA is condensed into chromatin fiber. That is the answer. Answer is option B. Histone proteins are the proteins which coil around the DNA in eukaryotes and form the rigid structure called as nucleosome. We studied. Then the positively charged end of the histones attract the negatively charged end of the DNA and leads to tight packing. This solenoid structure undergo further coiling to produce a chromatin fiber of 30 to 80 nanometer, and then a chromatid of 700 nanometer is formed. So the answer is, correct answer is option B. The DNA is condensed into chromatin fiber. The association of histone with the nucleosome indicate the DNA condensed into chromatin fiber. Answer will be option B.